All right, everybody, welcome back to the post-digestive, or maybe the digestive session. So, we're really getting a lot closer to the heart of the matter here in this session, which is around patient leadership. And I think we're going to hear different aspects of it from various perspectives, as parent, as patient, as funder of these efforts. And the focus here is what role can patients have beyond doing the important thing, which is raise awareness, contribute financially, participate in trials. But here we're asking a more pointed question. Traditionally, physician scientists are seen as the leaders of these kinds of efforts. What we've heard in our keynote is an example of how that might not be the case. And we've heard this from previous keynote speakers. And I believe our panel can speak quite clearly to this. Rather than having me introduce them and botching it, I've asked them in their introductory remarks to introduce themselves as they introduce their perspective on the topic of this panel. So without further ado, we're going to kick off with Farhad Imam, who has graciously agreed to fly all the way to share his views with us. Thank you.
Great, thanks, Zach. Thanks for the opportunity. It's a pleasure to get to be here and represent the Bill and Melinda Gates Foundation and speaking to this audience. I wanted to give you a little bit of an intro of my path to being at the foundation, and actually a lot of it was, many of the years were spent right across the street here learning to be a pediatrician and a neonatal intensive care doc, and then some of it across the Charles River over at the main Harvard campus, learning to do basic science research during my postdoc. And so the precision medicine that I used to do before I came to the foundation is this kind of precision medicine. And so most recently I was at UC San Diego before going to the foundation, and that patient over there on the left that you see is a critically ill neonate, not responding to standard therapy. And so the precision medicine that I did there was in combination with the Rady Genome Institute, which is a $150 million genome institute, and where we would sequence within less than a day the whole genome, about a third of the time get the smoking gun, know what that patient had, know what the genetic lesion was, and immediately be able to give the right drug that would stop that baby's seizures. About 2/3 of the time, we wouldn't get the smoking gun, we'd get a bunch of variants. And so then I would take those into my lab, and using animal models and CRISPR technology, which you'll hear about later from Editas, we would create patient-specific disease models and then see which one of those variants recapitulated the disease. And then furthermore, you could also go and see which drugs those would be susceptible to. So that was precision medicine in a high-income country. What I do now is precision medicine for these kind of patients. And so the question is, how do you reconcile the multi-million-dollar high-income country tech infrastructure for doing precision medicine in this kind of environment with this kind of patient demographic? You're going to get a few examples of amazing patient advocacy getting specific diseases on the map, on the radar. But how do we deliver the right medicine at the right time to the right patient in this scenario? For this patient, maybe the right medicine starts with clean water. Maybe it starts with vaccines. Maybe it starts with things that are basic public health infrastructure. And so precision public health, which is a new term that's kind of come up, is an interesting combination of the highest tech tools that we have at our disposal and the technologies that we're able to use here with the practicalities of delivering things at scale at a variety of different levels of sophistication of healthcare structure. So how we prioritize our map is the combination of the greatest areas of need and where we can have the greatest impact. To give you an example, the global health team so there's the infrastructure side, which is in global development. We talk about water, clean health, sanitation. Global health side, that's not my phone, I think. The global health side, HIV, TB, malaria, neglected tropical diseases, also some advocacy. But the bottom line is, if you look along those different columns, these are all different orphan or underrepresented diseases that have their advocates, like that advocate that you saw on the screen. And so really it's about addressing the gaps. And so in a country like this, the gaps are fewer and farther between. Moving to a lower resource setting, there are much wider gaps that we can already fill with existing solutions at the same time that we keep our eyes and ears open for all the other ones that we haven't yet seen and that people can bring to our attention from the grassroots, from the ground up. When we think about polio and malaria, there's goals like eradication. There's other diseases where our goal is reduced transmission or disease burden. You can move all the way down, actually, so our neglected tropical diseases program is by default neglected, and one of the ones that very much so traditional markets wouldn't go after. So we're trying to fill the gaps between where the market would go and where basic research and NIH-style public funding goes. And in between those two, there are not only gaps in this country and in this environment, but also in the low resource settings as well. So I'll just kind of put that as a slightly different spin to specific diseases that you will hear amazing advocates having been able to push forward. What we're trying to do is start with maybe people that, the basic solutions to problems that we don't have to wait for them to ask for. Very much like the first talk you heard this morning from Greg. As much as possible, let's try and take the burden off the patient from needing to drive the process. And he, in fact, started his work before he was a patient. And so very similarly, we're trying to start this work for all of us before any of us become the patient, because all of us may not be so fortunate as to have a dynamic, strong voice behind and getting the issue to the table, as you'll hear a few examples of today. So I'll start with that and sit on the panel. And Zach, are we going to wait for questions until everybody's spoken, or after everybody? Okay, thank you.
Thank you very much.
Good afternoon. Just try and find some space here. No, I think I'm good. Thank you, though. So I really appreciate the opportunity for being here today. I'm going to take you into a slightly different direction. And my name is Jocelyn Duff. I am the founder and executive director for a nonprofit called Cure CMT4J. It's an all-volunteer nonprofit dedicated to expediting therapeutic development for an ultra-rare disease called CMT4J. More importantly, I am mom to my two daughters, Tegan and Talia. Tegan is 15 and Talia is 12. So this is a picture of my daughter, Talia. This was from several years ago. And I'm here today really to share Talia's story with all of you. And unfortunately, I'm challenged with condensing about 9 years into just a few minutes, so I'll try to be very, very quick. On September 4th, 2015, my life changed forever. That sounds dramatic, I know. I was sitting at our kitchen table when our phone rang. I was home alone. My kids were at school, my husband was at work, and it was my day off. We had been on a diagnostic odyssey for the previous 6 years with Talia. She had gotten a flu-like illness when she was 3 and began to slide backwards in her motor milestones. She stopped walking. She began crawling upstairs and using her chin in this sort of funny tripod means of getting up there. And she stopped reaching her arms overhead to explore her world. Initially, she had been diagnosed with a treatable neuropathy, and we went down a long path of treatment with steroids, IVIG, plasmapheresis, cyclosporine. She actually got a little bit better with steroids and IVIG, but never quite back to her baseline, and weaker every time we tried to wean her off of the steroids. We had revisited her diagnosis many times. Previous genetic testing was unrevealing, but now we went back to look at a larger panel of genes. And so this call was a clinician telling me that Talia had tested positive for a mutation on the FIG4 gene and that it was a known disease-causing variant. She asked if I had any questions. Incredibly, I didn't. I honestly didn't know what I should ask. And like any good parent, I got off the phone and Googled the FIG4 mutation. I found a paper describing a disease called Charcot-Marie-Tooth 4J, or CMT4J. I scanned through the abstract and saw something that stopped me in my tracks. It was a comparison of CMT4J to another disease, ALS. In fact, a small percentage of patients with ALS also have a FIG4 mutation. I read on further to learn that CMT4J involved an autosomal recessive mutation on the FIG4 gene, on chromosome 6. It's almost always a peripheral neuropathy, rarely involving the CNS. And like many rare diseases, the age of onset and course of progression are highly variable. It could continue on as a rapid downward spiral or a longer, slower slog toward loss of muscle mass, increasing weakness in upper-lower extremities, joint instabilities and dislocations, frequent surgeries to stabilize those joints, severe scoliosis, progressive disability leading to quadriplegia, and premature death from respiratory compromise. And of course, there was no treatment or cure. I was devastated, heartbroken, angry, sad beyond belief, and terrified. I suddenly didn't know how much time we had with our daughter, but I knew I had to do something. And I quickly found myself in the world of rare diseases and ultra-rare diseases, with parents and patients finding and funding their own cures. The statistics of rare diseases are daunting. Over 7,000 rare diseases exist. 50% of rare diseases affect children, causing death or debilitating illness at a young age. 1 in 10 Americans is affected by a rare disease. 30 million people in the US and 350 million people worldwide have a rare disease. But the kicker for me was that 95% of all rare diseases currently have no approved treatment. And the interesting thing about some of these rare diseases is that some of them actually have a pretty good amount of bench science. In 2007, when the FIG4 mutation was first identified and given the disease name of CMT4J, only 22 cases had been identified throughout the world. By 2016, we actually knew a lot more about the FIG4 gene. There were mouse models, there were some postulated treatments, and we knew that overexpression of the FIG4 gene when replaced wasn't harmful. The incidence for disease-causing FIG4 mutations is now thought to be between 1 and 5 people per million, still exceedingly rare, and like so many other rare diseases, seemingly too rare to attract the attention of anyone to fund advancing the science. So when we learned of Talia's diagnosis, I dove into the existing research. I found who was working on the disease from a clinical standpoint and those who had done the bench science. I found researchers who had worked on drug development and translational medicine for other similar rare diseases, such as SMA and giant axonal neuropathy. To illustrate one of Greg's earlier points, the first researcher that I spoke with had been studying the FIG4 gene for nearly a decade, and yet he told me that he had never met anyone with CMT4J before. And then I discovered just how expensive it is not to bring a drug to market, but even just to get something through a small phase 1 clinical trial. Because after all, what was my ultimate goal but to try to halt this horrible disease in my child? We needed lots of money. We needed a nonprofit, and I needed lots of people to help me. I tapped friends, family, community members, people who possess skills that I didn't have, and thankfully, they all said yes. I was also fairly certain that gene therapy was the way to go, given that CMT4J involved a single gene mutation. And so I began to delve into the ever-evolving world of gene therapy, and I was quickly discovering just how expensive that world was and how bottlenecked the manufacturing industry is. But I put my head down and I remained laser focused, and so as a newly minted nonprofit, we started fundraising. We quickly raised some funds to put together the first ever symposium on CMT4J, and there all of our researchers remarkably agreed that gene therapy did indeed represent the best possible solution. We started preclinical work immediately and serendipitously fell under a grant from the NIH to the Jackson Laboratory. Fast forward 18 months later, and somehow we managed to raise more than $1.1 million with the help of our extraordinary community. We gained some national media attention through a viral video created by Talia's classmates and her teachers. We appeared not once, not twice, but 3 times on ABC World News with David Muir. And that got us our year-end goal of $1 million for 2017. We brought in over half of that through the magic of social media. That sweet video was shared widely. Thousands of donors contributed from every state in the US and 5 other countries. So now we've got some money in our pocket, we're in manufacturing queues, and have nearly completed our pre-IND. We're on course for a small phase 1 clinical trial in 2019, and have had a commercial partner actually reach out to help us get there. And our science actually couldn't have gone more smoothly. We have proof of concept using gene therapy with the AAV9 vector in our CMT4J mouse models at JAX, and we believe the use of AAV9 for CMT4J truly has the potential to advance treatments for other more common demyelinating diseases. So the pieces of our puzzle were first a set of signs and symptoms that were ultimately paired with a genetic mutation, a known disease-causing variant. And then it became about putting the therapeutic development pieces of the puzzle together, from bench science to approach to treatment or cure to preclinical work to a phase 1 trial. So the question, is patient leadership critical? I would argue yes, absolutely. Here was an ultra-rare disease, arguably one of the rarest of rares, known to affect 22 people in the whole world, with a surprising amount of basic science on a cellular level, but no one shouldering the effort toward a cure. Since establishing our foundation, we've gathered 16 individuals with CMT4J, a pretty big percentage of the presumed number of cases out there. There are likely hundreds of cases of CMT4J in the US alone, either misdiagnosed or undiagnosed, just as we were for so long. Stories like Talia's are becoming more and more commonplace. In some ways, this is obviously a good thing, in that rare and ultra-rare diseases are receiving more attention, and patients and parents recognize that they do indeed have a voice in creating and pushing treatments and cures. But in other ways, it highlights just how far we still have to go in delivering precision medicine to patients. The fact that parents are out there putting on bake sales, selling shoelaces, and making viral videos to gain attention for the funding of these cures is abysmal. At the same time, they likely have a medically fragile kiddo, and in all likelihood, their family is breaking apart from the stress and the heartache of it all. Someone else needs to be on the sidelines to help fund and further these treatments when patients get their diagnosis. Is precision medicine important? Absolutely. Without a diagnosis, without identifying the disease-causing mutation, we had nothing. But as this question has been asked so many times today, what do we do with that information if we don't have a treatment or a cure existing already? This is the critical piece. And how do we better support families after a diagnosis for which there is no known treatment? I wish you could meet Talia. You would fight for her too. She's smart, she's funny, she's empathetic, joyful, and so resilient. And she is a fighter. Talia needed a cure yesterday. She needed it 9 years ago when she first presented with symptoms. And people ask me, how is Talia? How is everything going? They're both extraordinarily difficult questions to answer. Every day that goes by is continued loss of function. Talia is now in a wheelchair. She's lost the use of her arms, and she now also has respiratory compromise. And while I know we've accomplished so much so quickly, it just isn't fast enough. Thank you.
Oh, thank you for sharing. I'm Stanley Nelson, and I'll try not to be emotional after your talk is very sweet. I'm the father as well. I'm father of a boy, and I do get emotional when I say this, even though I'm a physician and scientist and diagnosed many people with all sorts of terrible diseases over my career. My own son was diagnosed about 14 years ago with Duchenne muscular dystrophy. I'm reminded of it annually because where I live in LA, the jacarandas are in bloom, and so they rain these purple flowers down and create sort of a purple carpet around all the cars. If you're a car owner, you hate it, especially if you like your car, because it drops them all over. But it's a reminder annually of Dylan's diagnosis. My own background is I'm a pediatric oncologist by training. I was early into the genomics world and have built my career in genomics technology and informatics around that. And with Dylan's diagnosis, I actually left oncology and moved into rare disease for what I think is a bit of a bigger challenge in many ways. We are succeeding in pediatric oncology, and it felt like a lot of the models for how we move forward to actually substantially move that field forward were in place. Every child who came to UCLA was on a clinical trial. We managed dozens of clinical trials for every single child who walked through the door. If my child had been diagnosed with pediatric cancer, there'd have been a team of medical specialists around him, helping him with the multidisciplinary care that he needed. My child was diagnosed with Duchenne muscular dystrophy. Everybody in the room probably has heard of it. It's often in high school textbooks. So it's a rare disease, but it's a relatively common rare disease. So we didn't quite face the same obstacles that Talia did, but it was still shocking to me. As a pediatrician and an oncologist at UCLA, there was no organized care at UCLA, no organized care at Children's Hospital of LA, no organized care at Children's Hospital of Orange County, and none at Rady Children. So in our entire Southern California network, there's really no decent care for one of the most common genetic diseases of childhood. It appalled me, it embarrassed me, and it also felt like something that had to change. So one of our first tasks was actually we formed a Center for Duchenne muscular dystrophy and rebuilt what the clinical program looks like at UCLA. We facilitated the same happening at CHLA and at Rady's. And this enterprise has grown over the course of many years. So we now have a well-organized clinical care program. We have educational programs that actually teach an online course completely devoted to Duchenne muscular dystrophy as a paradigm of how do you make progress, how do you understand a rare genetic disease, and how do you bring and improve science training for undergraduates to encourage them to move into science, which is still actually a pretty big gap in our educational training. We don't teach science, we don't teach the joy of doing science early on in our professions. We don't really understand it until rather late. So we then have covered everything from research through clinic and then clinical trials as well. So in our journey through this, we've steadily been drawn in more and more to this issue of how do you translate knowledge in the basic science side to meaningful clinical efforts. This is going to be a little bit of a roundabout story, but I would argue that the field, when the DMD gene was first identified, right, actually just a block or so from here, by Lou Kunkel's lab and his students and postdocs, that identifying the disease gene, clearly identifying what the molecular cause of Duchenne was, so dominated the field that it felt like therapies were coming down the pike soon. As gene therapy, gene transfer became plausible to do, it also felt like the field was very focused on that one single goal, and that dominated the field in terms of therapeutics for Duchenne for well over a decade. In the last decade or so, we've pushed, and the field has pushed for a knowledge of, or a reckoning, sort of a recognition that indeed small gains are acceptable, making small progress, getting drugs that are not perfect, but are at least moving the dial forward, will indeed move the clinical course of Duchenne forward in a way that's actually meaningful. And that's actually pushed forward a number of different therapeutic areas. Actually, a number of the Duchenne therapeutic companies are here in your area in Cambridge. Two of them are Sarepta Therapeutics and Solid Biosciences, which are just across the river. I'm not sure what direction I'm pointing in, but one river that's around here. And these companies are devoted at the moment to Duchenne restoration strategies. So we've had a front row seat of watching companies take a rare disease drug, decide how to do clinical trials in a rare disease, do that in a way that allows them to gather the data to allow the FDA to give them approval. So we've been at advisory committees when the FDA has a difficult time deciding about a particular therapeutic. They'll have an AdCom. So an AdCom was held for the first cystic fibrosis drug, which actually did end up getting approved. It was also held for BioMarin for a drug called drisapersen for exon skipping. It was held for eteplirsen, again for exon skipping. And last year for PTC's drug for nonsense read-through. It's an enterprise that I would argue is broken. The FDA does not have the wherewithal or the means to meaningfully, scientifically review the data that's fundamentally behind rare disease research. If they can't do it well, thoughtfully, and with sort of rather strong rigor, not rigor on the side of rejecting drugs, but rigor on the side of permitting drugs that are reasonably likely to give you clinical benefits, to allow them to come into the clinical space, it's a broken system because we won't simply get the same answers out from these rare diseases as we will get out from much more common diseases. So watching this enterprise, clearly there needs to be a fundamental reckoning. 21st century cures is a step in the right direction, but it has to be implemented in a way that actually gets, and many people in the room will probably hate me saying this, is I think we do want a lower bar for getting meaningful therapeutics through to actual clinical practice. And I would prefer a system where we allow drugs out through the FDA with approval, with being able to pay for them by insurance companies, and then observe combinatorial effects of these drugs in real-life clinical practice. And that would be a far more meaningful enterprise. As eteplirsen was getting approved, which went through a controversial process, and even was the commentary for a number of people afterwards, it was viewed, and some of the comments were made, that if you approve drugs based on weak evidence, you'll never know that they're effective. And in the rare disease space, that's simply not true. So in the eteplirsen issue, if we took every single exon 51 skipping child in the US, it's about 13% of the patients, if indeed they walked longer than we'd have expected, which they do not do right now, which we know because of large database samples, that they would be indeed, the only difference is now they're post-eteplirsen approval versus the patients that we looked at 10 years ago were pre-eteplirsen approval. There's means that we can deal with these issues, and we don't have to deny kids sort of moving meaningful drug improvements forward. The other sort of string that I would comment on is one of the organizations that we work closely with, it's called Parent Project Muscular Dystrophy, started to collect online self-report data, so not involving a physician at all, allowing people to log on online and report their experience, what drugs are they taking, what nutraceuticals are they taking. Their idea was that this would be a way for them to be enrolled and brought into clinical trials. They also happened to ask, what drugs are you taking, and is your child walking? Is your child not walking? So in Duchenne muscular dystrophy, typically you'd lose the ability to walk at about age 10 years. And if you were having a meaningful drug that were slowing the rate of progression, you might expect that you'd lose ambulation later in life. So looking at these data, which are all directly online patient report data, there was a provocative set of data within this. And parents had responded to, can my child walk or not walk? Was my child on a steroid? If my child is on a steroid, which steroid? This particular story involves a drug that was not approved by the FDA in the US, deflazacort. So it's a steroid. It was advertised originally as a bone mineral sparing steroid from 30 plus years ago. It's been through various clinical trials. It's approved in Europe, so it's widely been given, but there's no great reason to ever give it to anybody because there's actually a lot of pretty good steroids out there. In the US, over the 80s and 90s, there's compelling data that prednisone in particular, given daily, slows the disease progression for Duchenne, and that's sort of overwhelmingly demonstrated now from a variety of means. One physician, particularly in Canada, sort of recognized that deflazacort had some features that might be better. So no sane scientist would look at this person and say, aha, he's definitely got it right, but he just plowed ahead and did it. And he convinced a number of other physicians that deflazacort might be more reasonable because of a variety of kind of subtle issues. And it turns out that a number of people in the US ultimately started to give their patients deflazacort, even though it's not approved in the US. You can do this, and this was news to me back when my son was diagnosed. You can do this through 90-day personal exemptions. So you can, as a physician, order a drug that's not marketed in the US. It can be shipped in legally with a prescription from overseas, and that patient can take the drug. So this has been going on for a decade in the US. So we could take this patient self-report data, and there's a scuttlebutt within any rare disease community. You get to meet most of the people that are in it. And within it, there's about a third of the patients were on deflazacort, about a third were on prednisone, and about a third weren't even taking a steroid at all because of diversity of opinions amongst physicians who still weren't convinced despite 2 decades of data. And that data showed that if you were taking deflazacort, you were associated with walking about 2 years longer than if you were taking prednisone. If you didn't take either, you walked 2 years shorter. Highly statistically significant because it was and is still the largest steroid study ever done for Duchenne, purely from patient online self-report data. And it appropriately predicted that deflazacort would be superior to prednisone. And indeed, since then, some clinical trials have been done, and that's the case. It's actually approved now as a drug for Duchenne. So there's 2 approved drugs for Duchenne through this enterprise. So I just wanted to mention that with the idea being, we're not going to be moving around in the dark for these diseases. And I think particularly in the rare disease space, we have to allow some way to move forward, but insist that we collect the data potentially forever on that. Thanks.
Good afternoon. My name is Carol Martin. I am with the Harvard Catalyst Program for Clinical Translational Research and the Program for Faculty Development and Diversity Inclusion. And I also work under the auspices of the Harvard Medical School Diversity, Inclusion, and Community Partnership. That's probably the longest title around here, and we have a lot of long titles. 2 years ago, I was diagnosed with stage 3 pancreatic cancer with squamous carcinoma, particularly virulent form of the diagnosis, and one that gave me projected lifespan of 1 to 2 months. I'm here today because of precision medicine. Let me check that and say precision drug treatment. And why I note the difference is because my view of precision medicine as a patient, as well as an administrator looking at this large entity from the inside out and the outside in, if you will, is based on the perspective that I think precision medicine from a patient perspective means the entire patient, not just a diseased organ. And unfortunately, if you think about medicine today, that is typically what your patient profile categorizes you as. Patient C, pancreatic cancer, inoperable tumor, and then squamous carcinoma. So each time I get a different designation, I go into a smaller and smaller box. And right now, because of the fact that I'm still here, I'm an N of 1. And for those of you into data, which is one of the things that I do very much in the course of my work, the N of 1 means my doctors are all surprised by the fact that I'm still here. My family is surprised by the fact that I'm still here. My coworkers are rather stunned by the fact that I was back at work and I'm still here. And I'll go into one other thing that I've done over the last year that still surprises me. So I want to just take you back a little bit to my initial diagnosis. Now, I mentioned the nature of my work. We're one of 62 clinical translational science centers around the country. We're the largest and the most complicated. If you think about most universities, there's the university, the graduate school, the medical school, and the hospital. Here at Harvard, we have all of those, and then we have all the teaching hospitals, and we have all the research centers. So we have 27 institutions as part of this one clinical translational science center in and of itself. And initially, I and all of my colleagues thought that my symptoms were based on stress. The work that I do is fairly stressful. The people that I work with can sometimes be stress-inducing. And as a consequence, it was no surprise that I was starting to slow down. As it turns out, I work in an old building just up the street, Longwood and Huntington Ave, and my office is on the 4th floor of a 4-floor walk-up. So when it was getting harder and harder for me to get up the stairs, I thought it was time to check in for another opinion. And it turns out that I went through a series of doctor visits and a series of doctors with varying degrees of assessment and diagnoses, none of which were pancreatic cancer. And it turns out that my initial breakthrough moment, intervention, if you will, came from one of my colleagues who, I run a summer program. My students are here right now, and I'm here. One of my colleagues who's been a mentor in our program for a number of years looked at me and said, you know, you don't look too good. How are you doing? And I said, not that well. And she said, well, have you been to see a doctor? And I said, yes. And she said, well, what did they say? And I said, well, they did this blood test, and they said my glucose level was off so much that they had them repeat the test. And she said, well, what was it? And I said, 400. Yeah, that was kind of the surprise I had as well. And so she said, well, did she have you get a glucose test kit? And I said, no, I don't know what that is. She said, here's what it is. Go up the street to CVS, go buy one, and then bring it back to my office. I'll set it up, and you'll have some data when you get to see an endocrinologist. So that's what she did. That's what I did. And when I went to see the endocrinologist, you know, one of the symptoms of pancreatic cancer, and once I got it, I thought, okay, people need to know what these are. So they include unexpected diabetes, type 2, back or abdominal pain, jaundice, pancreatitis, loss of appetite, nausea, vomiting, and unexplained weight loss. So I had everything except the jaundice, and still was not diagnosed. And I'm here. These are my colleagues. This is where I work. So I think about that fact for people not benefiting from this place and this network and these resources every day. And my goal as a consequence of being a patient on this side of pancreatic cancer is to make sure as many people know what those symptoms are, because they can be masked by so many other things. And as it turns out, once the diagnosis happened, the same person who said, you don't look good, said, do you want me to find you another doctor? And I said, yeah, you know, I think that would be a good idea. That day I had a new doctor. That next week I had the first appointment with her. And then the end of that week I had an upper MRI scheduled, went through that and thought, ooh, dodged a bullet, no breast cancer. The following week, I had the second MRI for the rest of me, and that was when they found the tumor. My treatment at that point went to warp speed. So the two bosses that I have, one who's the Dean for Diversity here at the medical school, and the other one who's actually the PI on our Clinical Translational Science Center, both acted as advocates for me at that point. Now, the PI is based at Dana-Farber. My boss was based at MGH, and so there was a little discussion, as you might imagine, about which hospitals I might end up receiving care from. And it turns out that my boss from MGH said, well, if you go to Dana-Farber, your doctor who found the disease will not have easy access to you. And so that was something that for me was the deal breaker. And I said, no, I'm going to stay here. I was at Beth Israel Hospital, and that's where I received all of my care. And once I made that decision, my treatment went to warp speed. So the first consult included the folks from surgery, the folks from oncology, the folks from radiology, a couple of other groups. And in all of those people, there were 9 doctors in the room and me and 2 nurses. And I was processing on the one hand, but on the other hand, most of the information was going over my head, mainly because it was so much at once and I was trying to take it all in, and at a certain level it just stopped. My boss, however, actually did know all the right questions to ask, and so she made sure that I had all of the information that I could then take and make the decisions that I needed for my next step care. I went to Dana-Farber as the second opinion, second decision place, and my other boss came to that consult. And the other person who came to the consult with the surgeon at Dana-Farber was the primary care doctor who diagnosed me originally. So, those kinds of things from a patient perspective gave me a sense that this woman really had my best interest and my ultimate health at heart. So I went through the chemotherapy, radiation therapy, and chemotherapy at the end with a drug called fluorophenox. Now, my oncologist has told me that fluorophenox is not normally used to treat pancreatic cancer by a number of oncologists because they're concerned about the toxicity of the drug. And I was given it in its most intense form and the maximum dosage over the course of treatment that I had. And they did the treatment that way, first chemo, then radiation, and then chemo. And what they found and what we found was that the precision medicine aspect of what they were doing, they adjusted the drugs every time I went in for treatment based on how I was presenting each time. And what my oncologist also told me to do was to think about the moments in my life that gave me joy. And it sounds simplistic, and it sounds outside of the realm of precision medicine conference, but actually was one of those things that I took to heart, and I actually did that. So he said, find things that give you joy. And I live in Harvard housing, and one of the things that they had was this program where they actually acknowledged somebody who made people feel welcome. Well, part of my job is to catalyze people. So I talk to people, I listen to their stories, I bring them together, I make connections for them. I do all of these kinds of things just as a function of who and how and what I operate and how I am. And so as a consequence of that, what they ended up doing was appointing me the Harvard hero for the housing office. And I got to go on Fenway Park Field, along with my colleagues from all the other universities 2 years ago, and they called us out on the field, called us by name, and they put our pictures up on the Jumbotron. So, of course, all of my guy friends wanted to know, how'd you get on the Jumbotron? Not that I'd done any of these other things, but it was one of those moments that, I probably wouldn't have done under ordinary circumstances, but because I had this latitude that I had no idea how long my life was going to last, I was going to live it. and I was going to let people know about what the symptoms of pancreatic cancer were. So one of the other things that I did while I was going through chemotherapy, right before I started radiation therapy, is I had been a very physically active person all my life, and I used to take part in the Tufts 10K, and in fact, up until that year that was my 13th year. That would have been my 14th year to do the Tufts 10K, and I asked my doctors if I could do the Tufts 10K that year, and they said, well, if you think you can do it, but you need to have somebody with you on the course. And so I said, okay. And my assistant actually came with me, and I said, I have just one request, that we not be the last people on the course. So we weren't, and we did. I finished. And then the very next day, I started radiation. Went through the course of radiation using CyberKnife and a very aggressive form of radiation therapy, even in that, and then started the chemo again right after that. I went through another 4 months of chemotherapy, and then I was done. I took about 2 months off past that and requested to come back to work. I went through Harvard accommodations, and they are very thorough, as you might imagine, in terms of looking at my space and looking at the tools that I needed to get back into doing my job. The nature of my work tends to be fairly cerebral, so the fact that I was on all of the post-cancer drugs, the opioids that are so much in the news, was problematic, and I asked my doctors if it were possible for me to wean myself off them. So they said, okay, in a staged fashion. And that's what we did. And so I'm back fully at work, and I'm back doing many of the things that I thought I would never have the opportunity to do with stage 3 pancreatic cancer and an inoperable tumor. So the one other thing that I did over the course of my treatment was, and this was again with my doctor's approval, to use alternate therapies and treatments to complement what I was getting in terms of the precision drugs that I was being treated with. So I did yoga. I did massage therapy. I have a masseuse who comes to my house every 2 weeks. She did it the entire time I was going through chemo, still does it. I started meditation this last year. And one of the things that our meditation teacher told us, it's a cancer group of patients, a variety of cancer types. And what he said was, here's the thing, you have the disease, you have the pain. One way you can deal with it is to dedicate it on behalf of something else, or someone else rather. And I should mention at this part that one of the significant side effects of fluorophenox is neuropathy. So when I tell you about this next thing, you'll wonder, okay, what was the connection and what thinking was going on between this one and that one? So the neuropathy in my feet is still very significant. And it became problematic about 6 months after I was back at work because I'm on the 4th floor of a 4-floor walk-up. No elevator. So the stairs were really problematic. And one of the things that I'm doing now with the university is actually talking to the accommodations office and HR about having accommodation intervention, not only when you first come back to work, but 3 months out, 6 months out, 9 months out, and maybe even a year out, depending on what are the conditions that patients are dealing with. The other thing that happened was the only surgery I had was eye surgery because pancreatic cancer also affects your vision, so I had that. And around this time, all of the hospitals were requesting people to take part as charity runners in the Boston Marathon. And since I'd had the surgery at Mass Eye and Ear, and I heard they had a pretty good team support program, I thought about doing the Boston Marathon. And with the thought from our meditation teacher running in my head that if you dedicate the pain on behalf of someone else instead of yourself, then it's not so insular and you're not thinking, oh my God, these really hurt. And why am I doing a marathon when my feet don't work properly? I decided to do it. So I trained for the marathon, and very methodically, like I do my work, and I did long training sessions on the weekend, and I did day-to-day in the gym in my building, and one of the things that I was doing was incremental extension of the distance on the weekend. And it turns out that something triggered for me to remember how to help my feet, because all the doctors were saying when I was saying the pain was really bad, gabapentin was the drug that they wanted me to take, and I'm the poster child for contraindications for drug side effects. So I didn't want to do that, and I remembered moleskin. I'm a hiker. I came here from Seattle, and I always had moleskin in my pack, and I thought, well, you know what, if I put that on, maybe that would help. And actually it did. And then the other thing that really helped and kept me going was the fact of the meditation and dedicating this on behalf of other patients. And so I undertook the training for the marathon with people supporting me, with the Harvard network supporting me and working with me to make sure I got my work done. And I actually ended up on the day that we had this year, Monsoon Monday, doing the marathon. It took me, my goal was 6 hours because that's when they cut off the clock. It took me 7 hours and 45 minutes. And I just want to mention that you go through, it's 26.2 miles. And the towns that you go through, Hopkinton, I had the note here somewhere. Hopkinton, Framingham, Natick, let's see, help me out, you guys. Thank you, Wellesley, Newton, yeah, Brookline, and then Boston. It took me 7 hours and 45 minutes. Once the clock goes off, they take the barriers away, so you're also dodging Boston traffic, and the policemen who are saying, no, wait, you have to wait, the cars are going through now. But we actually finished. I actually ended up finding a young woman who was by herself, and she was freaked out because her parents were concerned about her being there on the course by herself, and they said, you know, we're going to the same place, so we should go together. And this is what I got at the end.
Well, 7 hours. Well, that's one run that would kill me. So these are, I agree with you, Stan, these are very moving stories, but I feel almost obliged, and I think there's something bad in my character that's going to make me say something purposefully controversial. So when I look at this panel, we heard from Farhad at the beginning. And I imagine if he was to be asked, how many kids in Nigeria could you immunize and help for $400,000? You tell me it was a number that was considerably greater than one. At the same time, and I've been there in my own family, if $400,000 is going to give another year of life to someone I love, sounds like a bargain to me. So I'd like each of the panelists to just reflect on how does one do that trade-off, absolutely, and recognizing that your first duty is to the people you love. Nonetheless, we're also citizens of society. How do you do that balance? Stan, go for it.
Happy to start. So I think there's several things, and actually how I personally view the Duchenne space and a lot of the rare disease spaces. This is kind of the frontier where we're going to test genetic therapies. If CRISPR-Cas9 gene editing, the joke from Greg Simon earlier, if we're going to make success, this is the group of kids, this is a group of people that we're going to do it in, and we owe them a huge debt for their willingness to jump in, take these risks. And it's where we'll be developing the next realm of precision medicines. So to me, it's not necessarily that it needs to cost $400,000 to save a year, save a life, but it's more that this is where we are in terms of developing new therapeutics. This is the group that we can and should do it in, and it pays enormous benefits. It's a little bit along the same lines of diagnosing every child with a rare genetic disease. Is, just actually one of the other panelists said something that I strongly disagree with, is if you diagnose something and you can't do anything about it, what's the point? In genetic diseases, the diagnosis is profoundly important to every single person in the family, and giving them clarity of information, but also information for everybody else in the family who's going to otherwise factor in some risk of that same thing happening to them is profoundly important. So my attitude about this is we're in sort of a transition period where it's okay for these to cost individually a lot of money. I think as a society, we actually have to work to how do we do these types of profoundly important genetic manipulation experiments in humans in a way where the drugs don't need to cost $1 million at the end of it. And I think there's ways that we could get there in a way that would incentivize pharmaceuticals, incentivize families, interested people coming into it. Yeah.
Farhad, I can put you on the spot.
Sure. I mean, I think the answer is, as much as we can on all fronts. And the market works really well for certain places and certain populations, and has been tremendously successful in a lot of ways. Like the public sector and amazingly intelligent and dynamic advocates have done also incredibly well for places that the market hasn't worked. I think the challenge is there that if we view ourselves as a global society, what is the way that we map and address each of these things? And so, I'd mentioned the phrase precision public health and noticed a couple quizzical looks, which was the same look I gave the first time I heard it, but just to give a little bit more insight into an example of that. There's now an initiative in collaboration with the Institute for Health Metrics and Economics, IHME, where at the foundation we're looking at mapping down to the level of 5 kilometer by 5 kilometer resolution disease across South Asia and Sub-Saharan Africa. And the point of that, it's kind of a high resolution epidemiology in combination with next generation sequencing and really cutting edge mapping of the pathogen landscape, not only what are the usual players that are there that are causing disease, but what is the entirety of the viral, bacterial, parasitic space there, and do we need to think about how we map our policies, our public health policies, down to a finer level of resolution so that we can give maybe different antibiotics in certain regions because there's different bacteria causing disease in kids versus another region. If we have that knowledge, we can potentially deploy differential therapies and cures there. And to second your point on not all that knowledge is actionable yet. And so what if the cost of a monoclonal antibody or a novel vaccine were to come down? What if the cycle time of getting it through regulatory care also decreased? So we have also been funding platforms that empower all of those barriers to become lower and lower, so that providing custom, not state level, not country level or continent level, but rather regional cures, whether they're vaccines, biologics, small molecules, to the targeted populations that need them, becomes a reality. And so this is the idea of doing microbrew biologics or protein batches instead of needing multi-billion-dollar plants to do that. There's ways to do that now modularly for $5 to $10 million and make smaller, flexible types of drugs. And so all of these things, I think, are going to empower a landscape that becomes much more able to address smaller patient populations that traditional large pharma markets would not be attractive for.
Carol?
And I'm part of an entity that's all about collaboration on a high level, but at the same time, collaboration on a level that employs bringing in new minds, new thinking, as diverse a population to provide this care and to provide this type of research as the patients that we treat. And so the notion that anyone should be left off of the beneficiary of those resources once they become available. A lot of my students say they want to go to some of the countries that you identified to actually work with patients there. And the bulk of them want to stay here and work with patients in urban and rural settings. So, you know, there's that spectrum that we look sometimes outside that actually is very much present here. And the reality is that we need all hands on deck with this, and that's why I'm so encouraged by this conference and the rest of the work that Zach and his colleagues are doing here, and all of the people who are in this room, with the intent of finding a better outcome than we currently have.
One of my arguments being in the rare disease world is, you know, I always say, let's take a look at this disease and let's take a look at the amount of morbidity, the hospitalizations, the treatments, the therapies, and, you know, how quickly do we reach that $400,000? I'd say it's pretty quick. What we know about a lot of these precision medicine treatments that we're developing is that the faster that we can get to these patients, these kiddos, the better that these treatments are going to be. And, you know, the more that we can hone them, the quicker that we can get to diagnosis, I think the greater the ability that we can say, yes, the benefit, you know, far exceeds the cost of these drugs and developing these therapies.
So I'm going to ask one more question, and those of you who have questions, I recommend you start lining up because we're 12 minutes away from the break. So I'm going to start with you, Jocelyn, with regard to this question. We heard today, but we've heard on the news, a lot of concern about people trying therapies that may not be vetted in the traditional way, and that there's concern that there is potential for harm which is underappreciated, and that we're instead focusing, because of our desperation, on the potential for good. And I suspect in that discussion, which the right to try discussion is just one point on that spectrum, people are way overstating the contrasting views. I wonder what your take is from your personal perspective on what the balance is from a regulatory ethical perspective.
Well, I think obviously it's a very personal decision. I think developing a cure for your own child's disease is sort of part and parcel, a perfect example of weighing risk versus benefit. Here we don't even have a developed drug. We have an unknown. We have a viral vector, AAV9, that we feel, I think, pretty comfortable with at this point. It's been used in many other clinical trials and shown safety. But as a parent with a child with a life-ending disease and the possibility for a treatment where my child is in her disease course, the potential benefit for this therapy far outweighs any other perceived risk in my mind. And I think for other parents and patients out there who are weighing risk versus benefit for themselves, I do think it's a personal decision. Who handles the regulatory aspect of that? I'm not sure that I have the magical answer to that. And I'm not sure that we've made the right decision around that.
So, Farhad, I may be speaking, I certainly speak for ignorance, but I may or may not be wrong. You'll let me know. Which is, what I see is that there are trials, public health trials in Africa, such as HIV immunizations and Ebola vaccinations, which, because of differences in prevalence, would be seen as the wrong risk-benefit ratio to be done in this country. But they're not seen as on the wrong side of that cost-benefit risk ratio in Africa, for example. How do you see this discussion reflected in that public health precision medicine endeavor?
I think that's a great question, Zach. I mean, I think one of the scenarios I was talking over lunch about this, so just to bring it to a concrete example that hits this exact issue, we have a drug for birth asphyxia that we're working on that would potentially protect from children getting, children's brains getting injured and developing cerebral palsy. The regulatory path for this, depending on whether it's best given to the pregnant mom who's at a high risk for giving a child that's potentially asphyxiated versus giving it to a child that already has suffered that injury, and it could potentially moderate the extent to that and really help the healing. Two very different paths because of the risk profile ratio. On the one hand, there's a high-risk patient. She may not end up giving birth to a baby with a lot of complication and may end up not having a baby who's injured. And so what's your risk tolerance for giving a drug to a pregnant mother and an unborn fetus at the same time who may not end up having that disease versus what's your risk tolerance for giving that to a baby that's already been moderately or severely injured, is very likely to have lifelong debilitation, but may have incredibly large benefit from that if given right around the time of birth. And so I think those are two very different risk profiles and risk tolerances. We're in fact initiating a conversation with the FDA over that, of where you would, what are the requirements, what are the paths to these two things? These are two populations that have historically been incredibly difficult to do clinical trials in to begin with. Most clinical trials are done in adult men, and then there's so-called age de-escalation that gets brought down, and you usually don't even get to newborn babies, and yet as a neonatologist, I give all sorts of drugs to newborn babies who need it, who are critically ill, all off-label pretty much, none of which are actually tested in newborns, and some of which don't have the desired effect because babies aren't just small adults, they have different physiology. So how do we get to testing the drug that we want in the population that we want, and what's the risk tolerance of that population? Gosh, that's a really hard question, and I think we're struggling with it, but I don't think there's a one-size-fits-all answer.
And for Carol, I think you were already put through a decision process where you were asked to consider some pretty serious adverse events that may or may not help. And I wonder how that influences you around this whole view of regulatory braking versus, as in slowing down versus not. How does it make you think about these things?
Well, I guess the first thing that I would have to say to your point is it is such a personal decision and question, and the fact is that I'm here.
We're very happy.
That I'm alive, and 2 years ago that wasn't a decision. So, and the nature of my work is such that I'm always looking to help identify synergies for people who maybe can find that question, that tool, that new methodology that can advance science and medicine in ways that we currently can't. I remember when they were first talking about deciphering the genome, and then they said there was all this junk. They found these genes, but then there was all this junk. And I thought, no, that's not junk. It's just stuff that you can't translate yet. So I think we're sort of in that same moment with this, in that we've made such great strides in so many areas, but the ethical and the moral and the social and the humanist questions that we also have to weigh when we make these decisions. And as I was thinking about this, I think that location critically matters, because here we are in this place, in this state, with this concentration of medical centers, academic institutions. We have an informed and educated populace who votes and who is employed by many of these same entities. We have a venture capital community. We have a regulatory group. We have the insurance industry. We have pharma here, all working in the area of life science. So there are a number of people who have a vested interest in making this as dynamic and robust as possible. And I think with that, and then the philanthropic end, I don't want to leave that out, because if you think about all of the places that you've been to and see so many of the same names on so many of the buildings around, you know that that has a hand in it. And you know that people's outcomes benefit from those kinds of things. So I think that all of those sort of weigh into this, and as long as we have those many different groups weighing in on this, and it's not the arbitrary decision of one, I think we're in a better space.
So Stan, the particular disease area that you've focused on has been the subject of a lot of controversy. And parents, in particular, have been very active in letting the FDA know where they believe the chips fell. Could you reflect a bit on where you see these parents having contributed? You already hinted where you saw the FDA's role, and this is the hardest part of the question, can you imagine the risk, what's the worst thing that could have happened if you had been wrong?
Wrong about what?
About the risks of these drugs, for example.
Got it. Okay. So, like, for instance, the eteplirsen.
Yeah.
So, I think one of the things that's, is a cautionary tale is in terms, and I think this is where the FDA regulators appropriately land, is, even as physicians, when you hear a story from a patient, even a very nice patient, you sort of store it as, what? That's one story. That's good, right? It's really hard to store it in your physician brain. We do accumulate anecdotes, and we eventually move on those anecdotes, and actually some of the most brilliant physicians are great anecdote takers. I am not one of them. So I think the regulators often land on the side of, this is just blah, 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 meaningless information versus this is actually relevant information that could be factored in. So there was no, I know the popular impression is that parent advocacy indicating how great the drug was for their kids is what got it approved, and that's simply not the case. It was the fact that there was some evidence of dystrophin induction for the first time in history from an antisense oligonucleotide, and that coupled with a consistent signal showing within the clinical trial data, again done at one site, so lots of caveats to this, was sufficient to be reasonably likely to predict clinical benefit. I think it does. We can now look at long-term data. These same original 12 patients have now been on study drug for 6 continuous years. They're clearly doing better than the average. They were not a selected group at the beginning. We can look at a lot of those things. But I don't think we'll honestly know the end of the eteplirsen story until maybe eteplirsen's being phased out because there's something better, right? It may move that quickly. But 10 years from now, if the group of long-term administered eteplirsen kids are not shifted dramatically in their age at loss of ambulation, I'd be shocked based on all the data that exists right now. Are there risks that we would take in terms of exposing kids to drugs that the toxicity profile isn't fully well known? It is only 12 kids. I mean, there were some that were done in phase 1 trials in this, so it wasn't purely those kids. we assume it's a pretty high, good safety profile. Same could be said for Spinraza, but they're pretty small groups of kids. But again, I'd put my parent hat on. I think most parents who have a child with a lethal progressive genetic disease, how we think about that risk benefit is wildly different, and that's appropriate. And I think we're very willing to tolerate lots of risks.
So I think that's very important. People tend to treat this as if this is all other diseases. When you're faced with progressive one-way ticket.
Yeah, it's, I think for most of these diseases, the natural histories are actually quite well known. And others we don't know. And so I think, it's good to be kind of sanguine about it all.
So we have one question, and thank you. Uh-oh, so then in that case, definitely keep it short.
Although I hope it elicits a response. And it goes back to your first question. The 400,000 that might, in the third world, affect hundreds or thousands or hundreds of thousands of people versus the same 400,000 going to a very rare, ultra-rare disease even, where only a few individuals might be affected. If it were tax money, I think it's a real concern. But when it's private money, and this really goes opposite to where I usually come down on things, I don't think it's from the same pool. So I don't think money in the rare disease area would ever have gone to Africa or Southeast Asia or vice versa. And individuals, parents in particular, I think have a special right and need to protect their children. And if they can be fortunate enough to find small pockets of money to help them, that money would have gone to an app for some kind of social gain rather than to the rare disease. So I don't think it's
So that's actually a good question, and I apologize to the two gentlemen on my left. We're already over time, so we're not going to be able to take questions. Let me just add to the question. When I was a kid, we saw Saturn V go up, go to the moon, and people saying, why are we sending people to the moon? There's people starving. And I think there was a reasonable argument to be made that that money was not going to be sent to starving people. But how does one, is this gentleman making the point, a correct point or not?
Yeah, I'm not sure Tang fed a lot of starving children.
Yeah.
Sorry. I think it's a fair point. Actually, I think of it more as, it is a societal burden. For sure, we have a certain amount of money. We actually have a certain amount of income that comes into our society, and we spend it on a variety of things. And I'm absolutely changing how I intend to spend that money for all of society. And I intend to do the same thing at the NIH. I intend to do the same thing at the FDA, and I intend to get investors to shift what they're going to do. Part of the utility of that is to try to gain new knowledge in an important, key, absolutely fundamental new area. We're now manipulating the genome, right? The very first drug that just got approved, it's only been a couple of years, is an RNA therapeutic altering skipping in humans chronically. I mean, that's really stunning. It's the first. It's not the end. It's not a perfect drug. It's not well delivered. It doesn't get everywhere you want it to be, and it's not doing a particularly great job. But you got to start somewhere. I think as well as you start doing these things, having a drug that's approved because of a biomarker. So one of the most stunning things, sorry, this is long-winded, but one of the most stunning things at the AdCom was you had dystrophin restoration in a disease where you're genetically born without dystrophin was not a sufficient biomarker. The reason for that is it was at the very low end, but there's overwhelming evidence that the amount of dystrophin that you make is highly correlated with your disease severity. And we've actually had, up to the outcome, about 30 years worth of such data.
Yeah.
And yet it was really not embraced as sort of the core component of this that it should have been. So I think all these things it's worthwhile to shift what we invest societally into this rare disease, into new therapeutics, even very expensive therapeutics, because it's how we're going to move medicine forward. I think of it as an investment.
All right, I'm going to take this as an opportunity to thank the whole panel. And you have 10 minutes, only 10 minutes, be back.